So keep that in mind, and you know, that's why we have chapters assigned each week. But it should be quite logical and intuitive at this point in your as college students. Okay, so that's the answer to that question. If you have the time, and some students cram for the exam, some students read the chapters each week. You're all experts in this area, so I recommend you use whatever formula has given you the most success in the past. Okay. In terms of building neural networks and improving memory retention, it's probably wiser to accumulate information slowly over the week rather than cramming it last minute, which is easily forgotten. So, and if you do plan to become a scientist or a neurosurgeon, then you might want to, and you're fascinated by the subject, then by all means read the chapters each week. All right then, so any other questions? I'm here. I have a list of questions. Let me see, how many people do we have here? Participants, 31. So Abdullah, Andrew, Afrifa, and Lamisha. I got your emails, Andrew and Lamisha, about the scientist you would like to work with at Albert Einstein, and I already talked about your research potential with the scientist there, and I'm going to be working it out in the next week or two. So I will get back to you guys. If you don't hear back from me in 2 weeks, give me a friendly reminder, okay? Don't be shy. Pardon? Okay. Well, that's Andrew. Okay. Yes, neither of you seem shy, so just remind me if I forget. Okay. So I'm going to start the lecture then now, and then after my lecture tonight, which my lecture is shortened to accommodate the presentation of our first student presenter tonight, which she'll be presenting an Oliver Sacks story. And please, for your undergraduates, be aware of the structure of her Oliver Sacks presentation, because it's the correct structure for your presentation in terms of the topics covered and the number of slides. And how's everyone's connection tonight? Am I clear? Any problems out there?
Sounds good.
Okay, great. I can't see anybody, but I guess that's okay. Okay, great. All right, then, without further ado, let's move on to the lecture. And I'm going to assign these questions. And if you did not have a question last week, I would appreciate if you would volunteer this week to answer a question. And then if you can answer it correctly during my lecture, that's 1 point toward your first exam. And I've considered increasing the number of points per question, but that would have to happen in the next exam. Because this is the first time I'm experimenting with points towards an exam and for questions answered during a lecture. But I think I like it so far. So who would like to take the first question? What does the DBL stand for, and what does it do? And is that Naomi? Let me see.
Yes, I could have it. Naomi Lantigua.
Naomi Lantigua, did you have a question last week?
No, I didn't.
Okay, thank you. Number 2, what group of molecules are released from the ventral region of the blastula stage embryo that direct epidermal tissue development? Oh, Naomi's there, and one other person. Who else had just put their hand up?
With Abdul.
Abdul, did you have a question last week?
I did. So that was my question to you. Can we go again?
We have 33 students, and we had about 15
No problem.
last week. So I appreciate your enthusiasm, Abdul. Who else would like to answer the second question, please? Catherine? No, Catherine. Who's this?
Michelle.
Michelle? What's your last name, Michelle?
Gomez.
Okay, did you have a question last week?
No.
Okay, thank you. Number 3. Describe the process of convergent extension during neurulation. O'Hara? Molineros?
Yes.
Okay, O'Hara, Molineros. And number 4. Daniela Basave? Is that you? Is that you, Daniela Basave?
Yes.
And you didn't have a question last week, right?
No.
So your question is going to be to find the role of PCP protein in convergent extension.
Okay.
All right, so number 5, we have what regions does the zona limitans intrathalamica separate? And we have Dana Abda for that one. Is that right, Dana?
Yes, that's correct.
Fantastic. Number 6, which gene family exhibits spatially restricted expression and directs AP axis? Who's got that one? I need a name. Andrew, did you answer a question last week? Okay, you've got it, Andrew. Andrew Freefra. Protecting the country. Okay. Number 7, at which developing brain border is FGF8 active as a local organizer? I have. Is that, no, that's not Lucero Gill. He's not, he left the session, okay. What's the easiest way? I need a name, I can't see anyone's name. Number 7? Anyone? Someone? Danielle Preston. I have a Danielle Preston. And number 8, what structure is that? Famata Kajake. Is that right? Famata?
Yes.
Okay. Number 9, which molecule? So number 8 is what structure directs segmentation of the midbrain and hindbrain? Famata. Number 9 is which molecule expressed by the notochord directs dorsal ventral organization? I need a young scholar. Number 9. Number 9. Someone would like to answer number 9 during the lecture for participation. Can you hear me? Serena?
Professor, I didn't answer really good last week. I was wondering if I can make it up, if it's okay. I mean, nobody raised their hand, so.
Serena, you got your point last week, okay? And I'm going to let Sazine.
Okay, thank you, sorry.
Sazine John take it. Number 10, everyone. What transcription factor mutation leads to increased visual 1 cortex size and a condition called aniridia? Last but not least. Last. Number 10. Number 10. Shall I call on someone? Devin Dial, did you? Oh, who's this? Who's next? Who just volunteered?
I did.
What's your
Awa.
name? Awa. I know you. Okay, Awa. Thank you, everyone. Okay, now, for all of you, for the 10 of you who volunteered to answer the questions, you have to stop me on the slide after I describe it and say, you know, Professor, I'm ready to answer this question, okay? And then that's the way we can all, you know, sort of interact a little more online. I know you're out there watching and I'm here doing my best. All right, so before I move forward, does anyone have any answers for these questions before I describe them in slides, based on your reading of the textbook or past neurobiology courses or simple or basic biology courses? Would anyone like to? Yes? That is correct. It is a Hox gene. Who said that? Very good, Andrew. Okay, that's correct. Those are Hox genes. And we're going to go into more detail about that. Okay, any other questions that seem familiar? You may have seen before.
I took what is it called? Embryology, something like that. The first question is kind of like I know the answer. So the DVL stands for dorsal blaster per lip, and it basically induces like the formation of neural tissue in the exoderm.
Perfect. That's Henry.
Yes.
Very good, Henry. Thank you.
You're welcome.
Okay, very good. I am trying to get my screen to behave. All right then, without further ado, if I go back to this slide, let me give you the context for this lecture, and then we'll move into the material. This is neural induction and pattern formation. You know, last week we started with the big picture, the origin of the elements in the periodic table, the origin of protocells. Yeah, let me try to record this for you, Lucero Gill. Start recording. I'm going to give it a try, okay? So the title of this lecture is Neural Induction and Pattern Formation, and we're going to be building on the information we covered last week, which dealt with the origin of the elements in the periodic table, the origin of water on the surface of the Earth, the origin of protocells. Then we moved into the origin of prokaryotes, the origin of eukaryotes, the first neuron emerging as a sensory motor neuron or a mechanosensory neuron. And then we moved into the organization of the vertebrate nervous system. So we have to go back today and talk about the blueprints or the organizational structure that's put into place to build basic neurons and patterns of neurons as they develop and as they mature. So the title is Neural Induction and Pattern Formation. And we have to start with the beginning, which is, for us tonight, is going to be fertilization. Even prior to fertilization, where does basic organization begin? It begins in the oocyte, in the maternal oocyte. If you can look at this, let me use my pointer here. This is a maternal oocyte, and you can see polarization of mRNA and proteins in the single cell oocyte. The purple circle is the nucleus, and the green triangles inside will be DNA. And then you have red squares here at the dorsal region, and these kinds of smaller oval structures in the vegetal region. And you can see that they're color-coded. These are different types of mRNA that are localized to the vegetal region. And then you can see that there's FGF2 protein localized into the nucleus. And then there's ectodermal protein that's localized to the animal pole. So this first happens inside of a single cell. Some level of differentiation already exists in the unfertilized oocyte. Now, later at the 3-week stage in human development, there's going to be an incredible amount of organization and differentiation. And this is going to gastrula stage embryo. And it develops 3 germ layers primarily. Now, all these cells emerged from a single cell, but they all have unique expression patterns, and that's what gives them their unique identity. They all have the same genetic sequences that are the combination of the mother's and father's genetic material, but what differentiates them are the expression patterns, and the expression patterns of the individual cells are determined by the local environment and secreted molecules in the gastrula stage embryo. So if we look down here at this gastrula stage embryo, how's my pointer? Can everyone see my pointer on the screen?
Yes.
Yes.
Okay, great. So you're going to have an ectodermal layer. We're going to talk a lot about the ectodermal layer because that's where the neural and brain tissue comes from. Also in the ectodermal layer are skin cells of the epidermis and pigment cells. You'll have germ cells that emerge, which are sperm and egg. And if I drop down to beneath the gastrula, you're going to have endoderm, which are going to be lung cells, thyroid cells, and pancreatic cells. These are all considered endodermal tissue. And you'll have mesodermal tissue, or the mesodermal middle layer of the gastrula. That's going to lead to the differentiation of cardiac muscle, skeletal muscle, tubule cells of the kidney, red blood cells, and smooth muscle cells. So I want you to think back. It may be helpful to last week's talk when we talked about the central dogma, DNA leading to RNA, leading to protein. And it's really the central dogma that you want to keep in mind as the basic machinery driving gene expression patterns or gene expression profiles in individual cells, but also leading to the release of proteins or other secreted molecules or cell surface receptors that will lead to changes in local extracellular environments that will influence the identity of cells within those microenvironments. So let me keep moving on here. So this basic level of compartmentalization, and this is always fascinating too, just the initial polarization of the oocyte prior to differentiation. There's some central patterning involved in even at the single cell stage. This single cell patterning or compartmentalization of RNAs and proteins continues into adult neurons also, and it's described as zip codes now. Zip codes in adult neurons contain localizations or concentrations of different proteins or different RNA or other molecules or organelles as well. So this is there's still a lot to be discovered about how zip codes form or how polarization forms in individual cells. Moving forward, or moving back to the this blastula stage, there's a Mangold and Spearman organizer transplant experiment, which Henry alluded to already, that I would like you to be familiar with. And this happens within hours, this early blastula stage. I want to draw your attention to this panel A at the top. It says ventral to the left and dorsal to the right. This is the same, well, these are 2 separate embryos. One is a wild type embryo, or one is a wild type blastopore to the left, where my little digital finger is circling now. And in the wild type, you can see the dorsal blastopore lip, which is going to be central to the development of neural tissue. And in a wild type, this is a zebrafish development, then you have the single brain and single spinal cord developing. And then you have a central neural tube, somites, and a notochord. And these are going to lead to different aspects of the adult nervous system. If you move to the right, this Mangold and Spearman organizer transplant experiment simply took the dorsal blastopore lip from a wild type developing embryo and transplanted it into another wild type embryo. And that changed the neural architecture in the embryo receiving the transplant. And you can see what happened. The transplanted embryo was developed into two identical nervous systems, two brains and two spinal cords that were fused at the tail. And if you sliced through the mature animal, you could see that there were two primary axes. There's a neural tube at the anterior region and the neural tube at the dorsal region. So this is a process called twinning. And the question is, how would this happen? How does this work? And Henry, you're familiar with this. But I'd like the whole class to consider what could be happening with the transplantation of a dorsal blastopore lip that would lead to two brains and two spinal cords in the transplant recipient animal. Think about what we studied last week in terms of molecular biology and what you know from previous courses in development. Would anyone like to suggest what the dorsal blastopore lip does to change the nervous system in the transplanted animal? You can just speak up if you have any suggestions.
Is that like the organizer, or
It is an organizer. How would that work?
like it would like tell the cells that don't really have they don't really have a designated place to go, like it'll tell them what to be, basically, right?
And how does it tell the cells? What?
I have no idea.
Signal. Would it like signal like with something like some type of molecules to signal them?
That's right, Vanita. Signaling molecules, very good. And Abdul, did you have your hand up?
Yeah, I did. So I was going to say that it probably caused a change in the local environment of the cells that were in the original location before you had the transplantation. And so you probably had secretions, hormones that caused some sort of change in gene expression and led to the development of an alternative nervous system.
Fine. This basic molecular machinery I'm going to describe in detail in the next slide, but I want you to keep this in mind for the remainder of the talk tonight, which is that the dorsal blastopore lip is secreting molecules, proteins, for example. And the molecules that the dorsal blastopore lip that are releasing that are important are neuralizing agents are chordin, noggin, follistatin, cerebrus, and ADMP. Now, these are targeting cells that would otherwise become epidermal. And I'm going to walk you down to the bottom. Typically, the cells would become epidermal, and this is driven by the ventralizing center. This bone morphogenic protein, that is what BMP stands for. There's BMP4, BMP7, BMP2, BAMBI, and sizzled and tolloid. And in the absence of a second dorsal blastopore lip, the ventral region of the developing embryo would not develop a second brain, but would develop as a normal animal. So how does it work then at the molecular level? And just try to keep this in mind, because this is how essentially all the neurons in the central nervous system and peripheral nervous system acquire their identity, not from a dorsal blastopore lip, but by this cell-to-cell signaling, cell-to-cell contact, soluble factors, and localization. These cells are quite pluripotent or multipotent when they get, at this stage, they're quite pluripotent. That means they can become almost any cell in the body. As the nervous system develops, the cells acquire multipotency when they're within the nervous system. That means they develop the plasticity is reduced as the cells mature within a local environment. So these early neuralizing agents are going to work, and I'll walk you through this diagram here at the bottom. Chordin, noggin, follistatin, and cerebrus are extracellular antagonist ligands. That means they're simply proteins that antagonize the receptors that would normally bind bone morphogenic proteins. or they block the bone morphogenic proteins receptor or the proteins themselves. And here, if you follow this diagram at the bottom right, you'll see there's this large red T, large thick red T, and here you can see thalloid with a thin red T. The thickness of the lines in a cell signaling diagram like this indicate the strength of the signal. So the size of the arrows, for example, for BMP2, BMP4, and BMP7 binding this receptor this is quite large. Okay, let me know, Abdul, if it happens again. Everybody okay?
Professor, can you please talk if you got cut out when you were talking about the arrow?
Okay.
Can you please repeat that if you don't mind?
Absolutely, yes. And remind if it happens again, I may have to relocate again. But so the size of the symbols in this cell signaling diagram translate as the strength of the signal. A large green arrow from BMP2, 4, and 7 indicates that that's a strong signal, either a high concentration or a potent signal in its standard concentration. So I was indicating also that the large T, thick red T from chordin, nagostatin, follistatin, and cerebus indicates that that's a very potent inhibitor coming from chordin, noggin, follistatin, and cerebus that's blocking the BMP signaling. And if you follow this down, what the basic molecular signaling that occurs is first the BMP receptors are blocked, and that then leads, in the absence of this blocking, BMP27 would activate SMAD1, which would dimerize with SMAD4, and that would activate epidermal gene expression. But in the absence of this, the cells on the ventral pole are neuralized in the presence of the dorsal blastopore lip, which are secreting these antagonistic molecules. And that's how it works at the cell surface receptor and the second messenger signaling pathways and the nuclear level. If it says activation of target genes, that's happening at the nucleus. And if you go into bioinformatics or molecular biology, you're always going to want to follow this pathway from cell surface receptor or extracellular signaling, or there's extracellular concentration gradients, to cell surface receptor, to second messenger signaling, which is in the cytoplasm, to changes in transcription levels, which is going to be at the nuclear level. Keep this in mind. And then obviously these changes in nuclear transcription are going to lead to translation in the cytoplasm. And then the cells themselves will upregulate receptors to an extracellular signal, or they will upregulate molecules that they will release into the extracellular space in response to the extracellular signaling they're receiving. So it's a highly dynamic and plastic condition that leads to a cell acquiring a cell fate. Plastic in terms of it's a two-way street, and there's redundancy built into this system. You can see there's not just one pathway, but there are multiple pathways. There's even multiple inhibitors within the cytoplasm. And eukaryotic cells are built in a way that there are multiple signaling pathways that could lead to the same fate. And that is to ensure correct differentiation for the many millions of cells that lead to the properly organized central nervous system. Okay.
I have a question, Professor.
Yes.
It was the first question from the list that you gave.
Okay, go ahead. I'm sorry, I'm on the wrong slide. Go ahead, yes.
DBL stands for dorsal blastopore lip, and what it does is that it changes the neural architecture, meaning like it develops two identical brains and two identical spinal cord, which are the primary axes. And it acts as a signaling molecule that secretes the molecules to neuralize the targeting cells.
Okay, perfect. And that's Naomi?
Yes.
Okay, thank you, Naomi. Okay, Michelle, you have a question?
I have the answer for the second question.
Okay, go right ahead.
So the molecules that are released from the ventral region of the blastular stage embryo that directs epidermal tissue development are BMP4, BMP7, BMP2, BAMBI, scheduled and toloid. Correct?
Okay. Tell me the question one more time.
What group of molecules are released from the ventral region of the blastular stage embryo that direct epidermal tissue development?
Yeah. Okay. Very good. Thank you. Okay. We're going to move on now. Thank you both. I like this interactive work. Now, this is an interesting process. It's basic cellular mechanics. That's quite interesting. The question is, how would you go from a circular differentiating embryo or gastrular stage embryo? How would you go slightly more differentiated from a circular mass to an elongated mass with an anterior and posterior pole that's the familiar shape of the developing nervous system? And that occurs, the formation of a neural tube extends through a process called convergent extension. And the planar cell polarity protein organizes the cytoskeleton in these convergently extending cells along the medial to lateral axis. So let me show you what this looks like. Here I've typed in neurulation of the neural plate into a tube requires hinge points along the dorsal ventral axis produced in cells with apical constriction of actin cytoskeleton. So now when I scroll up and down, can you see the whole slide or just a portion of it? If I'm looking at this, can you see the whole slide or just
Nothing changed.
Nothing changes for you? Okay, wonderful. So then I would like to draw your attention to the diagram in panel A. You have an anterior, medial, lateral, and posterior region depicted. And what's happening here is convergent extension. It's simply cells from the lateral region of the developing embryo converge and move toward the center. That's the first step. And you can see there's an orange, blue, green, turquoise colored cell. And there are arrows pointing towards the center. They're converging toward the center. Sort of the way the teeth of a zipper will interdigitate, these cells are interdigitating. And as they interdigitate, the one direction that they can go is posteriorly. And so cell by cell, the neural tube develops in an anterior to posterior fashion by this process of convergent extension as cells interdigitate, thereby elongating the overall shape of the developing embryo as the tissue is neuralizing. And that's convergent extension. So this planar cell polarity protein orients the cell in this medial to lateral direction. It orients it, and then in a migratory fashion, the cells can move in the direction that the planar cell polarity protein has directed them to. First, the cytoskeleton reorients, then the cells follow. That's what the PCP protein is valuable for. And this first, the neural tube forms by hinge joint formation. Now here you have this developing structure, and you have the notochord in the center and somites on the right, and you have this medial and these two lateral regions. Now, there's not going to be an evagination if all the cells are cuboidal with a cuboidal cytoskeleton. The cells have to form a pyramidal or a triangular structure, and these are called hinge cells. These two red hinge cells are critical to the formation and closure of the neural tube, and they form by constriction of the actin cytoskeleton in that point where the neural tube is going to fold inward. If this is successful, you have a closed neural tube. And in the absence, we're going to look at what happens when there's a failure of neural tube closure. So this is basic neurobiology or basic developmental biology, showing you convergent extension pyramidal shaped cells and convergent enclosure of the neural tube. Now, I'm going to move on. Does anyone have a question to answer about this slide?
Yes, Professor, I do.
Okay, go ahead. Is that your Hera?
Okay, yes. So question number 3 says, describe the process of convergent extension during neurulation. So convergent extension is the process by which the tissue is restricted to converge or narrow along one axis and extend or elongate along a perpendicular axis. As you said, the cells from the lateral region in the graph, they converge to the center and they interdigitate and they elongate along the tissue. And the only way they can go is posterior. So that's what I got from it.
Fantastic. So your textbook will use an example of if you're in a movie theater, and if ever again we are in movie theaters and it's crowded, all the seats are filled, and the example is that those are cells in the prior to elongation. And the only way that the cells can get out is by funneling out of the exit down the hallway. That's convergent extension metaphorically, if that helps you to understand. Everyone has to form a side-by-side sort of orientation and then exit the theater. For you, it's in the text. 6 feet apart, obviously now. Thank you, Serena. That's very funny. Very good. Then that would be a disorganized nervous system if the cells couldn't touch each other. Someone had a question. Go ahead, please.
Yes, I'm answering question 4 about PCP.
Okay, go ahead. That's Daniela. Go ahead.
So PCP, which is planar cell polarity, is like they're different proteins that help with convergent extension by organizing the cytoskeleton. So like you described in figure A, how they line up, and basically what that protein does is help organize that along that axis.
Okay, very good. Thank you, Danielle. Okay, I'm going to move on to the next slide. Now, in the failure of correct neural tube closure, there are in our species, one of the familiar diseases is anencephaly, and that's a neural tube defect where the neural tube fails to close, resulting in the absence of a major portion of the brain and skull. And I give this to you future medical doctors because the previous slide is basic molecular biology or cellular biology, and this is I want to draw a connection between basic biology, molecular biology, and clinical or pathological conditions that you will encounter, and the significance of basic cell biology in terms of developing new treatments or understanding basic pathologies. So this basic pathology leads to, on the left, you see a fully developed newborn with an intact brain and an intact spinal cord. With anencephaly, this is failure of the neural tube to close, and there could be a normal skull line, but an underdeveloped brain, because the neural tube is going to form the brain and the spinal cord. If you look at a newborn, this is a clinical image of a newborn with anencephaly. There's a lack of you can see there's a lack of a large portion of the cortex and the skull. And these patients, from my understanding, will survive, but will have a great intellectual disability and can survive with the help of caretakers. So that shows the image of a newborn with that condition. So one of the molecular mechanisms involved in this anencephaly in humans is suppression of the transcription enhancer TEAD. And TEAD is involved in closure of the neural tube. So one reason that folic acid is prescribed for mothers planning to give birth is folic acid. Folic acid interacts with TEAD2 and significantly reduces the incidence of this defect. So here you have a very common prescription is folic acid. No, the spinal cord is not normal. I'm going to drop down to this here. So another form of anencephaly, there are many different forms, is spina bifida cystica. This is a failure of the neural tube lower down in the lumbar region that leads to herniation of the meninges as well as the spinal cord through the opening of the spinal canal. And if you look, this cross-section shows this yellow material bulging out of the back is neural tissue. It's spinal tissue, neural tissue. And that's bulging out, and sometimes it's under the surface of skin, and sometimes it can be surgically corrected. Here you could see another photographic image of what spina bifida cystica looks like. This is also a byproduct of failure of neural tube closure. So those are two clinical examples of what can happen at a molecular level that leads to an identifiable phenotype in a human pathology. Okay, so that I always like to make those connections for you. Moving on. Yes, Marianne? You have a question? Mariana? I need to hear you.
He wants to know why the neural tube does not close properly.
Well, it's a failure of convergent extension or a failure of the formation of the hinge joints. It can be any number of failures at the cellular and molecular level.
Is this something related to the gene, Professor?
Pardon?
Is that something related to any gene expression or not?
Yes, this transcription factor TEAD2 is involved in proper neural tube closure. And this is one of the genes that's implicated in this pathology.
Okay, thank you.
You mentioned, so is that like the red region in the other picture that you showed, or is it that it can happen in like other regions on the neural tube?
It can happen in other regions too. There are multiple types of anencephaly, and I'm showing you basically spina bifida cystica and a general anencephaly.
Okay.
So there would be different sort of expression patterns or genes implicated, perhaps in the different manifestations of the disorder. But one of the most sort of commonly prescribed preventative treatments is folic acid intake during pregnancy. So now you have a better idea of why someone would be asked to take folic acid as future doctors and scientists. Okay, I'm going to move on to the next slide, and we're going to jump to another section of this. I was just describing to you closure of the neural tube and correct patterning of the brain and spinal cord. Essential to this in most animal and insect species are Hox genes, Hox gene expression. These are quite penetrant and powerful genes, and they produce transcription factors that contain homeodomains, and those are DNA binding sites. What they do is regulate patterns of anatomical development in animals and fungi and plants. And to give you a sort of a diagram to help illustrate what this looks like, as you recall, all the cells in the given species are going to contain the same set of genes, but it's going to be the expression of these genes in this slide, or in terms of neural differentiation or localization, that will give cells individual identities. So here we're talking about compartmentalization or differentiation along the anterior to posterior axis of a fruit fly, of a Drosophila. And this Hox gene embedding, it doesn't mean that the Hox genes that are expressed, say, for example, in the lab region or here or in the antenna region, these PB genes that constitute the formation of the antenna are only expressed in this region. They are only expressed in this region, but they exist across the axis of the Drosophila. It's really just that the expression occurs in the head region. So the nesting of these genes in the anterior to posterior region looks like this. This is a color-coded. I'm going to move in a little closer. So you can see it says lab, PB, DFD, SER, ANTB, UBX, ABDA, and ABDB. That's abdomen A and abdomen B. And where you see these color matches between, say, ABDB, it's dark blue, and that's at the most posterior region of the abdomen. That means the morphology, the structure, the shape of that part of the Drosophila is determined by the expression of this ABD gene. And here, this antenna gene, the PB gene, the expression of the PB Hox gene leads to the growth of the antenna appendage on the head of the animal, on the head of the Drosophila. If you delete the PB gene, the Drosophila will not develop antenna. Similarly, if you express the PB gene in the developing fly embryo on its abdomen, an antenna will grow. And that is a potent and penetrant gene. So those studies have been done. If you want to see flies with antenna growing from their abdomen or wings growing from the anterior part of the head, those studies have all been done. And so these are ancient and extremely well-conserved genes across species, both vertebrate and invertebrate. So these, the next slide down, this is from your textbook. Also, you can look at for more detail. You have the fly. It shows you along the anterior to posterior axis. It says brain and nerve cord. It shows you the length along the body that this gene is expressed. So you have the LAB1. Lab is the Drosophila gene, and HOXB1 is the mouse version, the homolog. The mouse homolog is HOXB1, and the Drosophila homolog is Lab. The same gene, it's just expressed in different species and has a different name. So you can see PB is expressed across the axis. You have something like DFD that's expressed only in the brain region and in Drosophila. And you can see the SCR gene is expressed only in the head as well. So this is an expression map of these embedded Hox genes in fly. And this is a comparable or homologous map of the expression in mouse. And here you can see the mouse, the brain is broken down into rhombomeres, and I'm going to describe rhombomeres. They're going to develop into more differentiated and identifiable brain structures or cranial nerves. And then you have the spinal nerve. And so this is homologous, a comparison of homologous expression of embedded Hox genes that determine the identity of embryonic regions along the anterior to posterior axis. So these are turned on initially by a gradient. I haven't described to you yet signaling gradients that will pass across a developing embryo from anterior to posterior, but it happens in Drosophila. It happens in mouse. One of the most potent anterior to posterior signaling molecules is retinoic acid. This gradient will turn the Hox genes on. It'll be prior to the expression of Hox genes, you'll have less differentiation along the anterior to posterior axis. And retinoic acid is going to work on a cell receptor, second messenger signaling, nuclear transcription basis, the way the neuralizing agents worked in the DBL experiment. Retinoic acid is going to work this way too, but in a concentration-dependent manner. So at the anterior axis of the developing embryo, especially in fly, you're going to have a high concentration of retinoic acid. So if I come back here, there's going to be a high concentration of retinoic acid near the future fly brain, in the anterior portion of the developing embryo. And the source cells are going to be here that are going to be releasing retinoic acid. At the posterior end, there's going to be a decreased concentration of retinoic acid, a lower concentration. So what's happening is that gene expression sensitive to high concentrations of retinoic acid are going to be turned on in the head, while gene expression sensitive to low concentrations of retinoic acid are not going to be turned on. If you go back to the posterior end of the developing embryo, those genes that are sensitive to low levels of retinoic acid are going to be turned on, and that's going to lead to patterning of the posterior embryo. Okay, I said a lot, but this concentration-dependent gene expression is a very common mechanism in neural differentiation along the anterior-posterior axis, and you'll see along the dorsal to ventral axis of the developing neural tube. Anyone have any questions to answer from this slide? Okay, I'm going to move on. So once this initial patterning has been activated by retinoic acid signaling, which is called a global gradient, down to segmentalization of the developing neural tube on the anterior to posterior axis, then you'll have the differentiation of rhombomeres. And rhombomeres are transient axial segmentations that provide combined neuronal fate specification regions. Rhombomeres, they're combined neural fate specification regions. And if you look here, you have R1, R2, R3, R4, R5, R6, and R7 down to R8. These are going to develop into cranial nerves for this part of the discussion. And I'd like you to know what rhombomeres 2 and 3 form. Rhombomeres 2 and 3 are going to form trigeminal nerve. These are facial nerves or cranial nerves. Rhombomeres 4 and 5 are going to form facial nerve, the facial nerve. Rhombomeres 6 and 7 is going to form the glossopharyngeal nerve. And rhombomeres 7 and 8 are going to form the vagus nerve. And that's, you can see the Hox gene expression that's leading to the differentiation of these rhombomeres, and in a sort of domino effect, following differentiation of the rhombomeres, cranial nerves are going to form and emerge from this part of developing brainstem, and they're going to innervate organ tissue in the face or muscle tissue in the face, and modulate sensation or motor activity in the face. If you look at the right somatic motor neurons, these are going to be the trochlear. It's going to be emerging from R1, and the abducens is going to be emerging from R5 and R6, and the hypoglossal is going to be emerging from R8. So how these neurons are born, I'm going to show you on the next slide. Obviously, I want you to commit to memory global retinoic acid expression, or a global retinoic acid gradient leading to localized Hox gene expression, and then following localized Hox gene expression, there's going to be microdomain or localized changes in gene expression leading to neural fate determination based on local extracellular signaling or local cell-to-cell contacts. And once the initial identity of the neurons emerges, then there's going to be axogenesis. And axogenesis has its own signaling pathways that allows neurons born along the midline to radiate axons out to innervate the peripheral of the body. I'm going to move on. Does anyone have an answer to the question for this slide?
Professor?
Yes.
Yeah. Yes.
Go ahead.
Yeah. Yeah. So my question was which structure directs segmentation of the midbrain and hindbrain? So I know that there are several structures that regulate autonomic functions in the hindbrain, which are functions, things that we can't control, like sleep or motor and stuff like that. Also, there in the hindbrain, it consists of like pons and cerebellum and the medulla oblongata. For the midbrain, it also consists of the cerebral pedicle or the corpora quad, and also the cerebral aqueduct. So I know that another thing is that the brainstem also directs signals between the brain and the spinal cord.
Martha, hold on. This is I appreciate your enthusiasm. We're a few slides behind your slide. You're going to have a local organizer. This is going to be a single. I appreciate your description of brain structure, but you're going to see your slide when we get to it, and you'll be able to answer the question specifically.
Okay.
There's going to be a small piece of neural tissue, or that's going to be releasing a soluble molecule that's going to lead to that.
Okay.
Thing between those two brain regions. Okay. So I'm going to move on to the next slide. This is a diagram of what I've been alluding to or describing verbally for the past slide or two. And this is how we move from global gradients of something like retinoic acid, or in the developing fly embryo, there's another molecule called bicoid. These are global gradients that emerge at the anterior post, and they release there. And here you can see this happening here. So here at the, let's call the left region of this square of uniform cells, all these gray cells are cells with the same identity in this diagram. And this yellow triangle, at the very tip of the triangle, is the most concentrated, let's say, retinoic acid for this explanation. And then here, at the open end of the triangle, is the most diffused and lowest concentration of retinoic acid. So uniform sheet of cells, concentrated release of a retinoic acid gradient, and as it moves from anterior to posterior, the gradient becomes more diffused, and the cells in this posterior region are going to experience a lower concentration of the signaling gradient. And what would that mean? That would mean some cells are not going to be responding to very low levels of, some genes are not going to be activated by very low levels of retinoic acid, and some genes are only going to be activated by very high concentrations, and those are going to be in the anterior pole. So the next step would be a coarse pattern of transcription factor expression. This coarse pattern of transcription factor expression is going to lead to differentiation of early cells. Here, purple cells have an early fate emerging, and red cells have an early fate emerging. The red cells are, we can call them posterior cells, and the purple cells are anterior cells. The next is going to be cross repression, and number 3, between these blue and red cells. But this cross repression is going to be happening on a cell-to-cell level. These cells that are red are going to be contacting cells that are blue, and the cells will migrate into or out of that border. And by the time they get to this cell-to-cell contact-based migration, there's a sharp border will form. So these are just diagrams, but I'm going to give you examples next of what this looks like in terms of real neurons forming real borders, and then cells within these borders establishing unique identities. But this is a nice model to keep in mind as you think about going from a single cell to the highly complex and differentiated nervous system. If we drop down to number 5, then these boundary cells will form. In between the red and blue cells, the anterior and posterior cells, you have a middle ground where cells themselves will establish their own identity. And this is going to be by cell-to-cell contact. There are a number of proteins that act at the cell surface, cell receptor, cell ligand level, and I will describe those to you. But the molecular mechanisms are still very much the central dogma. whether there are some local extracellular gradients here, some local cell-to-cell signaling happening, that's molecules released from the red cells and the blue cells at the border, or whether it's physical contact between the cells, ligands expressed on the surface of one cell, binding to receptors on the surface of another cell. These signaling mechanisms are going to lead to changes in gene expression in the given cell population. And that change in gene expression is going to lead these multipotent cells, or these pluripotent cells, to multipotency, and then to terminal differentiation. As I mentioned, multipotent cells within the developing central nervous system have the potential to become any neuron in the developing central nervous system. And once these cells exit mitosis, these cells are all still actively dividing, you can assume. Once they exit mitosis, they acquire a terminal identity as a neuron. That's not happening at this stage, but later, if you drop down to 6, these boundary cells are expressing molecules, and then these boundary cells will lead to the migration of postmitotic cells. And these postmitotic cells are the final stage of neural development. They will migrate using chemotaxis or haptotaxis or chemorepulsion. These chemotaxis is the attraction of soluble molecules by a cell, or a cell that is attracted to molecules being secreted. So here you can see a purple cell, a posterior cell migrating to the former posterior region. That is not stochastic, or it's not a random phenomena. There are receptors expressed on the posterior cells that can detect molecules secreted by the anterior cells that are attractive. And this chemoattraction leads the cell to move into that region of the developing nervous system. So there are a series of signaling events that occur, and the postmitotic cells then can take up residence in the correct layer or the correct region of the developing central nervous system, and then sprout axons or form synaptic connections to lead to a more mature functional nervous system. Okay, does anyone have an answer to the question on this slide? Okay, I'm going to move on to the next slide. Now, these localizing centers lead to what's called early stage lineage restriction. And here, one of the early borders that forms is called the zona limitans intrathalamica. And here is the zona limitans intrathalamica here. This is the pre-zona limitans intrathalamica here, and it's in front of the proencephalon and the mesencephalon. And down here in red, you can see the notochord. The notochord releases its own gradient of sonic hedgehog. And you see the rhombomeres that we discussed in the previous slides, 1 through 7. And that process is called rhombencephalization. Now, you know some of the underlying mechanisms that leads to rhombencephalization, namely retinoic acid and Hox gene expression. And you have a structure here called the midbrain hindbrain border. And it's releasing a gradient of fibroblast growth factor number 8. And fibroblast growth factor 8, released from the midbrain hindbrain border, is going to differentiate the mesencephalon from the rhombomeres. Now, this is a local organizer. This larger this rougher architecture of rhombomeres, mesencephalon, and prosencephalon have formed based on global gradients, changes in gene expression, and the pattern we looked at in the previous slide. Now, local organizers are going to emerge. You can assume these are boundary cells from the previous slide. These local organizers are going to be releasing soluble molecules that will influence the identity of the cells neighboring in a concentration-dependent manner. So this pre-zona limitans intrathalamica, it says here it's absent of lunatic fringe gene expression, and is bordered posteriorly by the Iroquois Homebrox IRX3 protein, or IRX3. And that's shown in green cross-hatching. This is another level of organization, but still the Hox gene expression is involved. So if we move forward to a later stage of development, the zona limitans intrathalamica is formed, and that's here, this red triangular shape. This forms between the prethalamus and the thalamus. And here at the most anterior region of the developing central nervous system is the telencephalon, and behind this is the tectum, and behind that, even more posterior, is the cerebellum, and behind that is the medulla. The medulla is a brain. It's going to develop into an adult brainstem region that control heart rate, respiration, and other essential functions and develop in the adult animal. Moving on. Excuse me a second. Okay. Does anyone have any answers to questions on this slide?
I have a answer for question 7.
Okay, go ahead, Dana.
The question was, at which developing brain border is FGF8 active as a local organizer? And it's the midbrain and hindbrain boundary.
That's fine. Thank you.
Can I also have question number 5?
Okay.
The zonal limit. Dana.
Okay, so the last person was Danielle?
Yeah.
Okay, go ahead, Dana.
I have question number 5. So zona limitans intrathalamica separates the thalamus and the prethalamus.
Very good. And what protein is it releasing?
Sonic hedgehog.
That's correct. So sonic hedgehog is a canonical morphogenic protein. Canonical morphogenic protein in that it's involved in organization from early development into later development. And you'll see that sonic hedgehog is active in multiple tissue types and performs multiple functions. So here I'm introducing you to sonic hedgehog, but it's something that any developmental neurobiologist wants to be familiar with. And keep it in mind because we're going to discuss it again in the future. Move on to the next slide. Michelle, you have a question?
Yes. Can you repeat the answer for number 5, please?
Would that, Dana, did you have number 5? Would you like to repeat the answer, please, for Michelle?
So the zona limitans intrathalamica separates the thalamus and the prethalamus. It's between them.
Got it. Thank you.
It's also here, Michelle. And here on the diagram, you can see the zona limitans intrathalamica sits between the prethalamus and the thalamus. Okay. So if we look now at the neural tube ventral midline patterning based on the notochord, it's to the left. The notochord is a transient structure that's going to facilitate the organization of the neural tube on the dorsal to ventral axis. How does it facilitate cellular identity on the neural tube from a dorsal to ventral axis? That means top down. Here, up to this point, we've talked about organization of the neural tube from the anterior to posterior axis. Now we're looking at the tube itself, the neural tube itself, the dorsal being the top and the ventral being the bottom. At the bottom of the neural tube is the notochord. And here, the notochord is a little red dot that has the letter N on it. But the notochord is essential for allowing the formation of motor neurons. And here in yellow, you can see these motor neurons emerging from the ventral neural tube. And if you were to do a transplantation study, which this panel A is showing you, take the neural tube from a wild type embryo and transplant it into another wild type embryo, you would have something similar to the effect that you saw on the dorsal blastopore lip study, you would see something similar to twinning, but you would have the formation of, rather than 2 regions releasing motor neurons, you would have a 3rd region releasing motor neurons. And these motor neurons are going to innervate the body along the dorsal to ventral axis. Now, you are likely familiar with these motor neurons are going to emerge from the developing neural tube along the dorsal, along the anterior to posterior axis, and they're going to form the ganglia, spinal ganglia, that will innervate the body in a segmental fashion. The way we saw on the leech, the segmentalization of the central nervous system or the peripheral nervous system. This is the central nervous system. This segmentalization is in part going to be guided by notochord release of sonic hedgehog. And so at the bottom, you have a diagram showing you sonic hedgehog with it says SHH, that's abbreviation for sonic hedgehog. And you can see that there's an arrow pointing up. That arrow is leading to the ventral neural tube. The ventral neural tube, then the cells directly above the sonic hedgehog releasing notochord are going to express factors that will modulate transcription of cells medial to the tissue that the neural tube is influencing. And this change in medial expression in the ventral neural tube is going to lead to the birth of motor neurons. And these motor neuron cell bodies are going to, their fate will be determined in the ventral neural tube, and their axons will emerge through a process called axogenesis, which will then innervate different parts of the body. So that's the story. That is the molecule. There's sonic hedgehog here on the previous slide, between the thalamus and the prethalamus, and here sonic hedgehog release from the notochord is driving the differentiation of motor neurons. So you may ask, what happens if we deleted sonic hedgehog from the developing neural tube? You would see an immense loss of neural tissue forming or motor neurons forming, if not a total loss of motor neurons forming at the ventral tube. Okay, I'm moving on. Does anyone have an answer or question on this slide? Okay, I'm going to move on. Now, here's another more descriptive diagram of the slide we just looked at. There are BMPs being released from the roof plate. And sonic hedgehog is responsible for generating interneurons. And these interneurons are labeled as V1 to V3. And motor neurons in half of the spinal cord. BMPs are responsible for generating interneurons, and these are labeled as D1 to D6. So this is interesting because it's the first time that we're going to quantify how much sonic hedgehog is necessary to give a neuron its identity. And you may or may not be familiar with this level of concentration, but here you have the floor plate where the notochord was releasing sonic hedgehog, and you can see this dark red leads to a light pink. That's a visualization of the diffusion and decrease in concentration of sonic hedgehog. And here you can see these motor neurons, V3, MN, V2, V1, and VO. And to the right of this, for the first time, which is interesting, because there's such subtle differences in the concentration of sonic hedgehog at the nanomole level that can change its fate. So if we look, you can see that the first concentration that a cell reacts to is 0.5 nanomole. And 0.5 nanomole is going to give birth to the VO motor neuron. 0.5 nanomole is such a small amount. It's very hard to dilute down to that level to stimulate cells. We work in the lab using 100 nanomoles, or a micromole of these signaling molecules to change neuron behavior. In particular, we work with these types of signaling molecules or morphogenic proteins to see if we can drive the migration of neurons in my lab. So 1 nanomole of sonic hedgehog is going to give birth to V2. 2 nanomoles is going to give birth to MN. 3 nanomoles is going to give birth to V3. And the floor plate neurons are going to emerge from the highest concentration, which is going to be 4 nanomoles. That's a lot for a single cell or a population of cells in the notochord to emit. Now, keep in mind where the sonic hedgehog is coming from. The individual cells in the notochord are the factories through the central dogma that are producing sonic hedgehog SR, sonic hedgehog RNA, which is being translated into protein, which is likely being embedded or transformed into its final form into the Golgi apparatus. From the Golgi, from the endoplasmic reticulum to the Golgi apparatus, these vesicles. Okay, Serena. Where did we stop, Serena? What was the last thing I was describing, Serena?
When you started talking about the Golgi?
Okay, great. That's just a microsecond ago.
Yeah. It was only a few seconds that it broke.
So I'm trying to convey you the amazing process of a population of cells in the neural tube working as sonic hedgehog factories, going through transcription, translation, packaging of sonic hedgehog into vesicles from the Golgi, transporting it down microtubule highways to their dorsal surface. From the dorsal surface of the cells in the notochord, these vesicles have to be released and then have to diffuse up into the developing neural tube in a very highly synchronized manner and controlled manner to give birth to these individual neuron populations at the nanomolar level. So that kind of synchronization and timing and precise molecular machinery, it's still there's still so much to be understood about how this works in terms of molecular biology and neuroscience. This complexity is there's still a lot to be deciphered. So if you're aspiring scientists, I give to you these problems to pique your curiosity. And let me move on. Does anyone have a question to answer on this slide?
Yes, I do. It was question 9. It says which molecule expressed from the notochord directs TV organization. And from what I heard from you, I think it's the sonic hedgehog and the BMP that responsible to generate interneurons and which directs the fate of cells. That's it.
Okay, very good. Now, Famata, are you out there?
Yes, I was just trying to unmute.
I think you might.
Yeah. So, what So also, what I understood was also the rhombomeres that you mentioned that deals with segmentation within the hindbrain and the midbrain. Yeah. Sorry, what was that?
Can you repeat the question you're answering?
Oh, which structure directs segmentation of the midbrain and hindbrain?
And that structure is?
The MHB.
That's correct.
Yeah.
Very good. Thank you. Famata?
Yes, because I was also confused because when I read in the book also it also spoke a little bit about the rhombomeres as the segmentation of the hindbrain.
Yes.
In terms of like patterning stuff like that.
Yes. The rhombomeres are involved in patterning. They're each going to generate individual populations of neurons that will have unique identities depending on their localization.
Okay.
Okay.
I'm
Very
sorry,
good.
but what was number 9?
Sazine, we have a question about your question. Can you repeat your question and answer?
Sure. Let me just find the question again. Okay. Number 9 was which molecule expressed from the notochord directs PV organization, and it was the sonic hedgehog and BMP.
Okay, it's just sonic hedgehog released from the notochord, okay?
Okay, got it.
Very good. Nice effort on everyone's part. And let me move on to some transcription factors involved in, following sonic hedgehog signaling. Then there are transcription factors. Transcription factors bind to DNA and activate expression of particular genes. So you can imagine sonic hedgehog targeting neurons in the ventral neural tube. The first stage of gene expression could be the expression of transcription factors. These transcription factors would then turn on the right combination of genes, whether it's 4 nanomole, 3 nanomole, or 2 nanomole of sonic hedgehog activating this expression pattern. Likely, the concentrations of sonic hedgehog are going to be activating different combinations of transcription factors that will then lead to different combinations of genes being expressed, which would give neurons unique identities. And obviously, after our RNA transcription, you have the identity of a neuron emerging from its protein translation and localization of proteins. In particular, localization of proteins into the cell membrane. These proteins that are embedded in the cell membrane can be transmitter receptors, voltage regulated receptors that give neurons unique properties, sensitivity to different receptors or responsiveness to incoming signals. So this diagram, follow me through this and I'll show you sonic hedgehog signaling from the ventral pole induces expression of transcription factor NKX 2.2, NKX 6.1, and represses expression of PAX6 and DBX2. And here you can see this diagram showing you another concentration gradient, except this time instead of the sonic hedgehog protein, these are concentration gradients within a cell of the transcription factor. And as this moves, you have mutual repression in the center, and then the emergence of progenitor regions that constitute early neural fate or fate determination. So here you see a little more detail at the transcriptional level, what gives birth to the VO, V1, V2, MN, V3 neural progenitor cells that will then have a unique identity. And I think that's right. You can see that sonic hedgehog in this lower region is going to inhibit PAX6 and DBX2 expression and stimulate or drive the expression of NKX 2.2 and NKX 6.1. So granted, this is a highly simplified diagram of the cell signaling that occurs in response to sonic hedgehog stimulation from the notochord. So the last slide for tonight is regulation of neocortical patterns, and some of the morphogenic proteins we talked about already are going to appear. This is later in development when the cortex is emerging and differentiating. You can see there's an anterior neural ridge, and from the anterior neural ridge, you have the release of the FGF8 protein. FGF8 stands for fibroblast growth factor 8. And you can be sure that the cells closest to the anterior neural ridge are going to receive the highest concentration of FGF8, and the cells furthest away within within a detectable limit are going to receive the lowest concentration of FGF8. And that's going to pattern surface cell identity along the neocortex. Now, in the medial ganglionic eminence in blue here, you're going to have the release of sonic hedgehog. And sonic hedgehog here at the medial ganglionic eminence is going to be released. And that's also going to be working in a concentration-dependent manner. And lastly, at the cortical hem, you're going to have bone morphogenic proteins released. These are the same proteins that are released in the blastula stage embryo. Now they're being expressed again in a different neighborhood, in different zip codes, and rather than differentiating between neural and epidermal, or helping to drive that process, their concentration is going to drive the identity of individual neurons in the cortex. So the transcription factors that are activated by these morphogenic proteins are EMX2 or PAX6. And EMX2 is abbreviated as it is empty spiracles homebox 2, and it encodes a homebox containing transcription factor that is clearly involved in patterning the neocortex into defined functional areas. And now you can think back on the homunculus. This is where the patterning, the functional architecture of the neocortex emerges that gives birth to the final somatosensory mapping that the homunculus describes. So if you have a wild type animal, meaning normal expression of the morphogenic proteins in this slide, then you're going to have well-segmented functional regions, motor, somatosensory, and visual. M1 is motor, S1 is somatosensory, and V1 is visual cortex. If you have an EMX2 mutant where it's deleted, then you're going to have a disproportionate allocation of neocortical territory to different regions. You're going to have a larger motor cortex, a larger somatosensory cortex, and a smaller visual cortex. The same is true if you have a PAX6 mutant. Not the same types of mislocation of territory, neural territory, but a different mislocation of territory. You'll have an extremely large visual cortex, a very small somatosensory cortex, and a minute motor cortex. So if you have a loss of gene function, a loss of, say, PAX6 gene function, there's a pathology in humans called aniridia. This is a result of a PAX6 mutation, and it leads to what is called iris hypoplasia. And here to the left, you can see a healthy, developed iris, and here you can see hypoplasia due to a PAX6 mutation, and it shows that the iris no longer has the correct morphology or symmetry. And you could be sure that vision in this eye would be decreased or absent. So that's aniridia, the result of a PAX6 mutation. Again, we're moving from basic biology, cell biology, and morphogenic gradient signaling, patterning of the cortex, and one example of what happens when there's a malfunction or a mutation in one of the canonical transcription factors, PAX6. Someone has a question to answer on this slide.
Yeah, that's me. It says, what transcription factor mutation leads to increased V1 cortex size and aniridia? So basically, like you mentioned, if there's a mutation in the PAX6, you would have an increased visual cortex, which could also lead to this condition that you mentioned, aniridia, which means your iris is either incomplete or partial.
Excellent. Okay, now, has every question been answered?
Yes. Oh, actually, no. Nobody answered number 6.
Okay. I believe that is Andrew Afrifa's question. Okay. Does everyone get the answer to that question? Very good. Okay, now I'm going to, let me see if I can stop recording. I guess that will record until the end. So now we can.
Professor, I don't mind.
Okay. Oh, actually, I'm gonna stop. So I can clear my throat. It's the first time I've ever recorded a lecture in 10 years. I'll have to look at it to find ways I can improve. So now we're going to move on to one of the most exciting parts of the course, a student presentation. Did you email me your presentation, Benita?
I did.